Hello and welcome to Life at PALS. Welcome back to part 2 of the AS level structured questions paper 2 for biology. So we are having our second screencast on that paper. In screencast 1, in lesson 1, we have completed question 1 and 2, where both questions were completed, and question 1 was about these food molecules here, or different types of molecules, these biological molecules, their bonds and structures and functions. And question 2 was about mitosis and the cell cycle, and chromosomes and DNA. So these two are completed. You can check out the first lesson for these two. Question 3 is about enzymes, and this is what we will have a closer look at in this lesson. So let's dive right in. Question 3. The enzyme glucose 6-phosphate dehydrogenase, G6PD, is composed of two identical polypeptide chains. Students investigated the activity of two forms of this enzyme, J and K, at different concentrations of substrate. K is a form of the enzyme that results from a mutation that changes one amino acid in the polypeptide. The results are shown in figure 3.1, which we see below. We can see in this graph that with changing substrate concentration, the rate of reaction will change for a constant enzyme concentration. And we can see that when we increase the concentration of substrate, the rate of reaction increases until at higher concentrations of substrate, the rate increases at a slower rate, in both cases until we reach a maximum rate at 60 micromole per minute for J and 50 micromole per minute for K. Use figure 3.1 to complete table 3.1 by stating the Vmax and the Km for enzymes J and K and the units of Vmax and Km. So this is assumed knowledge. You need to know what Vmax and Km stands for. Vmax stands for the, not volume, the maximum velocity of the reaction, which means the highest possible rate of the reaction for a given enzyme concentration. Let's have a closer look at what this means, and Km as well. Km is the so-called Michaelis-Menten constant. So let's revise a little bit what these two variables stand for. So Vmax, as substrate concentration increases, the initial rate of reaction also increases. Again, this is only what we would expect. The more substrate molecules there are around, the more often an enzyme's active site can bind with one. However, if we go on increasing substrate concentration, keeping the enzyme concentration constant, so not adding more enzyme, only adding more substrate, there comes a point where every enzyme's active site is working continuously. If more substrate is added, the enzyme simply cannot work faster. The substrate molecules are effectively queuing up for an active site to become vacant. And that means at this point, the enzyme for this enzyme concentration is working at its maximum possible rate, which is known as Vmax, and V in this case stands for velocity, so the maximum velocity. Actually, let's complete this part of the table before we go into Km. So maximum velocity, fairly easy. Maximum rate of reaction, we just go back to the graph, which I have here on the left side now, and we check for both enzymes J and K, where is the maximum rate. The maximum rate is where the curve flattens, and that's if we add more substrate, that's when the substrate molecules are queuing up basically. So we have the maximum rate for J here, and the maximum rate for K is here. So 60 micromole per minute and 50 micromole per minute. Obviously, unit should be clear as well. Unit of Vmax is micromole per minute. And we have 60 and 50. Okay, Km, the Michaelis-Menten constant. What is Km? Another useful value can be obtained from the double reciprocal plot, namely the Michaelis-Menten constant. The Michaelis-Menten constant is the substrate concentration at which an enzyme works at half its maximum rate, half Vmax. At this point, half the active sites of the enzyme are occupied by substrate. The higher the affinity of the enzyme for the substrate, the lower the substrate concentration needed for this to happen. Thus, the Michaelis-Menten constant is a measure of the affinity of the enzyme for its substrate. The higher the affinity, the lower the Michaelis-Menten constant, and the quicker the reaction will proceed to its maximum rate. Although the maximum rate itself is not affected by the Michaelis-Menten constant, and Vmax and Kam therefore provide two different ways comparing the efficiency of different enzymes. Vmax gives information about the maximum rate of reaction that is possible, so though not necessarily the rate under cell conditions, while Km measures the affinity of the enzyme for the substrate. The higher the affinity, the more likely the product will be formed when a substrate molecule enters the active site, rather than substrate simply leaving the active site again before a reaction takes place. These two aspects of efficiency are rather like using the maximum speed and acceleration to measure the efficiency of a car. So in this case, maximum speed would be Vmax and the acceleration would be Km. So Km gives us an idea about the affinity of the enzyme, how well does it work when it's not fully occupied, specifically at half Vmax. That means half of the active sites are vacant, they're free, and that means the lower Km, the lower the Michaelis-Menten constant, the lower the substrate concentration at which we reach half Vmax, the more effective the higher the affinity of the enzyme for that specific substrate. So we can find the Km using two methods. I don't want to go into the first method because we don't need it in this question, which a bit more complicated, which involves taking the reciprocal of Vmax and substrate concentration and then make a plot of -1 over Km, and that's the x-intercept. This is how we can find Km here on this plot. More easy for us is this method, so let's go over. Now we said, okay, Km relates to the substrate concentration that is needed to reach half Vmax, half of the maximum rate, and to find that, we already know for J our maximum rate is 60, so we halve that, we go down to 30, and we find the corresponding x value for that, which is 0.2. We do the same for K. For K, our half Vmax is 25, 50 divided by 2. 25 is here, so we go over And we can see we land on 0.2 substrate concentration as well. Since Km is a measure of substrate concentration, we use the same unit. So in this case, we will have millimole per decimeter cube. And in both cases, this is 0.2. Okay, with reference to figure 3.1 and table 3.1, describe the effect of the mutation on the activity of the enzyme and suggest an explanation for this effect. Okay, what can we see? We can see that with reference to figure 3.1, at any substrate concentration, no matter where we are on this graph, the rate of reaction of enzyme K is slower than that of enzyme J. So at all substrate concentrations, the rate of K is slower than the rate of J. What else can we say? We should describe, okay, now we know that K is always slower than J, doesn't matter for which substrate concentration. What else is an important difference here? We can see that the rates diverge further. So we can see at a substrate concentration of 0.2, K has a rate of 25 and micromole per minute, and J has a rate of 30 micromole per minute. So we have a difference of 5 micromoles per minute here. If we go further up in the concentration of the substrate, for example to 0.6, we can see that our difference in rate of reaction has increased. Now, what's that? 46, and yeah, it's getting bigger and easier up here. We can see that at a substrate concentration of 1.6 or 1.4 already, we have a difference of 10 micromole per minute. So with increasing substrate concentration, the difference between the rate of J and K is increasing. In questions like these, we always get free marks by stating specific values to support our descriptions that we have given here. So we can use the two differences that I have mentioned already. At concentration 0.2, too lazy to write the unit now. The concentration of 0.2 millimole per decimeter cube. Difference in rate is 5 at concentration 1.4. The difference in rate is 10 micromole per minute. So that should be 3 points. We need one more point, and the last point we can score by suggesting an explanation for this effect. And here we have several options. So all boils down to the mutation that we have and one amino acid that has been changed. Where did the mutation probably happen? Eventually at the active site. Mutation may have affected the active site and this means that the affinity of K is lower, or it's a bit different. Actually, that works. So the affinity of enzyme K is lower than J, which means we have a slower rate. Of reaction in K. All right, let's move on to part B. In certain conditions, G6PD may also exist as 4 identical polypeptide chains rather than 2 identical polypeptide chains. Explain why both of these types of G6PD have all 4 levels of protein structure. That shouldn't be too difficult. Both have 2 or more individual polypeptide chains where each chain has primary, secondary, and tertiary structure. Two or more chains combining and binding to form the final enzyme is the quaternary structure. Okay, so yeah, we can have a quick look at enzyme structures. Why not? Primary structure of enzymes, of proteins in general, is the amino acid sequence, just the connected amino acids forming a chain. Then the secondary structure are alpha helices and beta pleated sheets. Two very common structures where when neighboring amino acids form hydrogen bonds to get into these structures. The tertiary structure are specific bonds that can form between amino acids that are not neighboring and far away from each other by looping and bending the amino acid chain and bringing two amino acids different positions close to each other, and these two amino acids may then form an interaction. And there we have probably the most prominent ones to mention are the disulfide bonds and we have hydrogen bonding here. We may have ionic bonding, where basic parts and acidic parts of an amino acid or rests of amino acids interact with each other, one losing a hydrogen, the other gaining a hydrogen, forming positive, negative ion, and they interact with each other. And as well, nonpolar interactions. Some amino acids may be nonpolar, they tend to stick to each other as well, avoiding the water they are surrounded by, because these nonpolar regions of amino acids are hydrophobic. Then we have the quaternary structure, and this is where we have fully folded polypeptide chains, different polypeptide chains that come together and bind to each other, forming the final working protein. Most prominent example for quaternary structure protein, probably hemoglobin. We have the 4 subgroups, the 4 polypeptide chains. Only when they've all 4 combined together, we have a working hemoglobin protein. Okay, this was question 3, a bit longer. Next time we will have a look at question 4 in the next class, or maybe question 4 and 5. Let's see how complex the questions will be. So today in this lesson only one question, question 3. I hope I could help you with your understanding of enzymes and specifically the bit confusing parts, Vmax and Km, the Michaelis-Menten Constant. I hope you understand it a little bit better now. And as well, typical question of describing a graph, how to approach it, and never forget the 3 points that you can score in these questions by just stating some important values from the graph that are relevant to the description that you have given beforehand. Okay, thank you very much for joining. If you liked the video, please consider to subscribe to our YouTube channel. This increases our reach and we can help more people like you to prepare for the exams. See you next time for question 4 and 5.